Thank you so much for having me. Thanks to the organizers. It's a really fun event given the sort of the breadth of the audience. There's the math, the biology, and the physics. And I'm realizing that I'm actually in this audience count as a biologist, which I think my bio friends would find hilarious, but I'll try to sort of fill those shoes. Yes, so my goal is to try and, the invitation was to talk about some open questions that would speak to this multidisciplinary audience. That's what I'll try to do. So I'll try to sort of maybe say less, but hopefully be understood, and pitch some questions that I believe are important, which biologists, under some definition of biologist, would find useful, that I think this community has all the tools for addressing. And, some of them I think might be even as simple that we could just like lock ourselves in a room in the afternoon today and just like do them, maybe. So that'll be the discussion part, which is totally optional, but that's before lunch for some of the open questions that I'll try to pitch at you guys. Anyway, okay, so I'm a theorist. I work in microbial. Very brief context. If you may know that most of the life on our planet is microbial. Microbes live in these very complex ecosystems, incredibly diverse, hundreds of coexisting species. And there's a long history of the interface of this field with statistical physics. Why? Basically because the goal is to predict community behavior without knowing all the details, right? And that's a very stat physics-y goal. And I want to say that, microbial ecology needs large N thinking, because there's basically no way we can predict anything about them by understanding 300 coupled differential equations. We know that, right? So there's just kind of no other way. Like, this has to be part of the solution. And
300 wrong equations.
300 wrong equations, exactly. Thank you, Daniel. Absolutely true. And yet, I'll put a yet here, the biology community working on these problems and the mathematical and physics community working on similar problems, there's very limited crosstalk between these communities. Somebody told me an anecdote that some ecologist tweeted back when tweets were a thing, that like, oh my God, I just discovered, turns out that, physicists work on ecology. They write all these papers and they publish in physics journals. Isn't that amazing? I've never heard of that. So why is that? And to sort of briefly talk about it, this is meant for kind of general context. So content will start on this board. Then I'm all open for questions. This is kind of like briefly for maybe opinions. But okay, so where's the source of predictive power coming from? And in statistical physics, if I were to reduce this to two ideas, which is a great thing to attempt in this audience, but I'll try. Point one is that large N leads to emergent laws and behaviors in a way that's actually incredibly general, because a lot of that is just because high-dimensional geometry is a very weird thing. And we heard about this, right? We had this example of a sphere that, all of the volume of a ball is at the surface, and all of the surface is actually next to a diameter. And that's not just some cute, fun fact of high-dimensional geometry. That is actually the exact same statement that when you put two gases together, they thermalize at the same temperature, right? Because kinetic energy, the velocities live on the high-dimensional sphere. So there's something very general about this. And there's another second deep idea, which is that, often this replacement, instead of studying this system, which is hard, we think of it as, turns out that typical properties of an ensemble of systems are sometimes possible to characterize. And many properties, if it's true that some properties of a system do not depend on all the details, then they should be shared by that ensemble. And if we choose the ensemble right, we may be able to predict something, understand something about our system as well. Okay, And that's very important, very deep ideas that were incredibly successful in many contexts of complex systems. But if I now think about things about real microbial ecosystems, the interesting fact is that increasingly the empirical examples that we see where we're actually able to make some prediction at the level of a community that did not depend on exactly who was there, right? So precisely the kind of predictions that we want, in cases where we can track them down to where they come from, they happen to come from facts of physiology, biochemistry, metabolism. Now, let me give you a couple examples. So, for example, carbon metabolism. Now, as a biologist in the room, I can tell you that carbon metabolism looks like this. There is, a circle, and then there's this little tail sticking out about how, various carbon compounds are transformed, and some bugs run it this way, some bugs run it that way. And it so happens that, you can do it either or, right?
There's some arrow coming in for input.
There are many arrows coming in, but that's the important thing is that you can't run the same tail in both directions at once, right? And that's called gluconeogenic and glycolytic metabolism. But because of that, across, if you assemble ecosystems on some carbon source and the relative fraction of bugs, there's a natural kind of division into two broad classes, and the ratio of those two broad classes is reproducible and predictable because of a thing like that. Okay, just one example. Here's another example which I personally find very fun. So it's a fact that I learned from Sepa, who's one of our speakers tomorrow, I think. So nitrite, particular compound, is toxic at low pH. Who knew? Right? Okay. That's a fact of life. Now, because of that, it so happens that when you think about a process called denitrification, which is happening in the soil, now this is nitrite, and in acidic conditions, if it accumulates, that's toxic. So if there are any red bugs running this reaction, they're inhibited by this accumulation of nitrite. And therefore, the blue bugs who run this reaction need to catch up and use up the nitrite to relieve this inhibition. Basically, and at low pH, therefore, community needs to find a way to run this process so that this process doesn't accumulate, this metabolite doesn't accumulate. And you make certain predictions out of that. Importantly, that because it's a fact of life, it doesn't depend on exactly who the red bug is, exactly who the blue bug is. That's a prediction that doesn't depend on all the details, which is exactly the kind of thing we want. But you see the issue. These are the kinds of factors that are absent in typical models, kind of statistical physics models of ecology, right? So these predictions come from facts of physiology, biochemistry, metabolism, and not just large N. And I put this in quotes because calling something just large N is a very, incredibly underselling the power of large N, right? But there are these extra factors that shape real communities. And what I kind of want to argue is that
are those sort of loosely like in statistical physics, there's energy which is conserved and numbers which there's various things that are conserved which change the behavior.
Exactly.
But there's a small number.
So there's absolutely no reason why this is incompatible with a large N.
But typically the number of things, these kinds of constraints and things are much smaller than the total number of players in the community.
Yes. So, but if there's some constraint that is affecting shaped by shared physiological constraint. If there's some constraint that are affecting everybody, like for example this one, right, then this may have consequences. And I think this is absolutely accessible to the statistical physics treatment. You know, the fact that carbon metabolism exists and it's, you know, has something is known about it, what effect does that have in large N limit? Right? That is absolutely perfectly statistical physics question if we only can find a way to ask it. And I think that this will open up a lot of questions that would be very directly of interest to a large swath of biological community as well, right? So it's not like there are many questions that are good questions that don't directly engage with us, but if we found a way to engage with this, that would open up new questions. That's what I'm saying. So what we need, or at least what would be really fun to be able to do, is to marry the large N thinking with at least a caricature of something that looks like these physiology, metabolism, etc. And so, again, absolutely no reason for this to be incompatible. So all we need to do is just find a way to ask the statistical physics question here, which is, of course, how do we turn physiology or metabolism into JIJ? This was meant to be a joke. Sorry.
It's true.
And so the idea that I want to kind of describe today is that physiology and metabolism we can think of them as interactions, but these are interactions of traits. So, for example, acetate and glucose are not typically co-utilized by the same bacterium because one of them corresponds to running this branch in one direction and the other in the other direction. So the sort of property of eating this resource and eating that resource don't go well together. They don't like each other, and I can put that into a JJ, and that can be my caricature of something like a metabolic fact.
Sorry, what is the N?
What is the N?
Yeah, what is being counted?
Yeah, so here,
the number of species.
Number of species or number of resources, or, yeah, so I'm about to define a specific model.
Assumes the definition of species.
Oh, yeah. So I'll be talking about...
We go to the large N.
Second?
It requires that you are clear as to what is a species.
Yes, I'll be talking about phenotypes in a second, where I'll be describing organisms in a community by what they do. And therefore, you know, that will be my microscopic description, and I'll have a large number of such things, right? I very much like the question of what exactly is a species, but here I won't have to ask it because I'll be just counting different phenotypes.
I mean, for this purpose, the right answer is who cares?
Say again?
Who cares whether you call it a species or not?
Exactly. Very good. That's exactly right. So, and generally I'll be looking for predict functional community behavior. This is what I'll be want to do. By functional, I mean things like, you know, uptake of some resource by a community, and they're not, so these are functional as opposed to compositional questions, like what is the distribution of abundances of a typical species, right? I want to be asking this question, and that very naturally kind of hides away the questions of like, well, exactly how do I count species or things like that. Okay.
They may be related because the definition of species may depend on function.
Yes. Yes. And if I care about everything, I need to know everything, and therefore I need to describe every single phenotype down to exactly how many flagella it has and what transcriptional state it has, in which case maybe it's 15 trillion individuals, right? And not 100 species.
Can I push back a little on that statement as someone who's tried to do this a lot? I think the trickiness is actually doing the function. You can write models like you wrote, where you collapse all the diversity and think about the function.
Mm-hmm.
That's pretty easy. You can write models where you think about the diversity, but don't think about the function. The whole thing that's tricky is to get both at the same time.
I
So
agree.
doing one or the other is pretty.
I agree, and I'll be thinking about function. For today, I'm just going to be thinking about function. Like, yes, that's a very intentional choice. There's a lot of people who work on diversity, and they're excellent questions in that space. I want to.
Alone is pretty easy, I would say.
Say again?
Either one alone is pretty easy, I would say.
Yes, that's what I'm hoping for, and that's why I'm pitching you this, because we had some things where I'm sure we could do some awesome analytics like in a couple hours, and then it'll be great. Okay, so basically, I want to turn all this. This was my pitch, right? This is now where the content starts. I want to describe to you a specific model that we like to play with, which I want to say kind of implements something like this, and therefore opens some interesting questions. And of course, it's one model, right? And we can discuss its faults, but that will also make this discussion very concrete. Sounds good? Okay, so the approach. So again, the goal is to marry large N thinking with a notion of physiological or metabolic constraints. And my approach will be to think about shared constraints in a large N framework of an ecological model. And specifically, which one will I pick? I will pick what's called a consumer resource model. It's a shorthand for species competing for resources with evolution. Why with evolution? I'll explain. Okay. So now I'll just write the equations of a model because it's very simple and it'll give us a lot of concreteness. So I'll think about organisms described by traits, which I'll label I, which go from 1 to some large number L. And I'll think of this as trait as can consume a resource I. And so I'll think of them actually as just binary. My organisms will be, which I'll call species, though maybe I should call them phenotypes or something. My species will be this, I'll call them sigma mu I. These will be, they're describing my phenotype, and they're just binary vectors of length L, which is which resources my phenotype can consume. Yeah.
Scale of L with N?
Say again?
Do you assume any scale of L with N?
L and yes, L is, so number of species will be of order L, but actually in all of what I'll show you, I will not actually do any analytics. I apologize. That's what I hope people will show me how to do. I'll describe some questions and show some simulations. But yes, I mean, a number of coexisting species will be at most L at equilibrium, and I expect them to be sort of scaling together and both going to infinity. Okay, so what are my dynamics of species? It's the simplest thing I can write. The growth rate of a species per capita is determined by what? So sum over all resources. If I can eat them, then I get the benefit from that resource Ri. It's available at some concentration, right? And then I'll have this is how much resources I collected. This is how much some energy needs that I have. And if I collected more than I needed, I can grow. And if I don't collect enough, I can't grow, I'll be dying. Very straightforward.
So this is just 0, 1, right? Or
sigma mu i's are just 0 and 1. I mean, that's for convenience of actually thinking about evolution. There's a lot of analytics you can do assuming that they're just Gaussian with some variance. It doesn't matter. Okay. So these are the resources. And what are the resources? So what's the dynamics of resources? Well, I'll be thinking about what biologists call a chemostat, which is this sort of the setup where it's a vat where flow resources at some rate and there's some outflow, and that maintains some community in the vat. So my resources arrive at some rate from outside. They are taken up by the community. So sum over all the species that eat the resource sigma mu I and mu Ri, that's the community, that's depletion of the resource. This whole thing here, I'll call this phi I, that's the uptake flux of the resource I, which is a possibly interesting functional observable. And also, my resources are depleted. There's some outflow that just depletes the resources. Make sense? Very
all the species eat the same at the same speed or something.
Yeah. So you can put in different coefficients in various places. That's not an innocuous choice, because as I drew it like this, there's a Lyapunov function on these dynamics. I will we can discuss that. I am very aware of that being a very strong assumption, which here, for the kind of questions I'll be asking, I think that's not a problem. And in fact, it may simplify some of the analytics. So I think of that as a benefit. But you can put in different...
Highly non-generic.
It's highly non-generic, absolutely. I'm proposing...
Sigma appears in both places.
Yeah. I'm thinking of this as something like, an integrable model that allows you some insight into a problem, even though it's very special. That's what makes it integrable. Here, for some questions, existence of that kind of function may be very helpful. And if we should always check in simulations if whatever we find survives, adding, for example, different yields here, which have both index mu and I, and that will kill the that.
I guess for the purpose of this workshop, it means there's a landscape, while usually there isn't.
Yes, but I will show you in a second that actually here this will be a snowscape, not a landscape, even here, and that is explicitly there to make Daniel happier. Okay, does that make sense? That's basically the simplest model you could possibly write about organisms who consume resources, and therefore resources are depleted by organisms. That's it. Now, okay, where are the constraints? I advertised to you that all of this is going to be about some caricature of metabolism and physiology. I want constraints, shared constraints. So constraints appear here. I didn't define to you what is this chi mu, which is how costly is it to be phenotype mu, right? And I'll say that how costly is it? Well, there's some baseline cost. There is, I don't know, making your DNA or whatever. There's a cost maybe per trait for carrying each trait, maybe it costs you something. But crucially, traits interact. So some traits go together well, and some traits go together poorly. Okay? So if I want to maintain, so here is a fact of biochemistry. The enzyme nitrogenase, which is an enzyme that fixes nitrogen from the atmosphere, is killed by oxygen. Like molecular oxygen destroys that enzyme, makes it non-functional. Which means that if I'm an organism who wants to do both things, I want to be able to fix nitrogen, and I want to be able to run aerobic metabolism dependent on oxygen. This is a difficult thing to do. It is costly because I need to invest in some infrastructure to compartmentalize those two processes away from each other. Different bugs do this differently, but everybody needs to contend with this issue. That if you try to just naively do it, they will kill each other. So this is an example of two traits that if I want to carry both of those traits, my cost is higher. That make sense? And there I can also give you examples of like traits that are synergistic that go well together. And importantly, my assumption here will be that notice this thing does not have an index mu. It is an interaction between traits, which is a fact of biochemistry or metabolism and applies equally to everybody. And that's what makes this an interaction that doesn't it's not an interaction between species 1 and species 2, which in the large and limit, who cares? Is an interaction between everybody who carries trait 1 and everybody who carries trait 2, which does not.
And you need the J to be symmetric, right?
Here, yes, J is symmetric.
Sorry, the Jij is a function also of mu. There is an index mu, right?
So it could have been, and that's what I'm saying. I will say that's an assumption of my model, that this is meant to implement the world, the biochemistry of my world. The fact that nitrogenase is killed by oxygen, I mean, technically somebody could have maybe evolved an enzyme that doesn't have this property. It so happens that, maybe there's some universal reason why that's hard. So there are plenty of models where we think about, interactions that are all idiosyncratic, that are all property of exactly which species is 3 and species 17, all of their quirks shape their interactions. This is the opposite limit of interactions are determined only by the traits they carry, and this JJ is the same for an elephant and an E. coli. But this is just a microbial model.
Intracellular interactions.
Yeah.
As against the interactions via the resources, which are
Exactly.
extracellular.
Exactly. Exactly. And so really quickly, I'll also, so when I said with evolution, what is evolution here plus evolution, which is that I allow bit flips 0 to 1, and I'll call that evolution, but I should be calling this changes of strategy. Like the bugs in my community may change what they're doing, and I'll often be calling this mutations just for habit, but the better, so I should put quotes here, because the biological version of that is, things turn on a pathway, turn off a pathway, they change what they do. That doesn't have to be evolution. But here I'll be calling it evolution. This defines the model. Now let me situate you in the context of sort of what this model is. How does it talk to other models in the field?
So
The
can
capital
I ask you?
RI, this thing coming from outside
Yeah.
is just whatever you want.
Yes, this is this is all in a chemostat. There's some influxes of resources, which I'm God. I set them to whatever I want.
In practice, this is the thing the experimentalists can control.
Yes, exactly. This is what experimentalists control. This will actually come back and be important. This is determined by the system. I don't get to control directly that. But of course, who survives, who grows, who dies is determined by this equation, which involves RIs and not big R, right? So this is the sense, and I'm running ahead a little bit. You can think of this as kind of like a fitness landscape, right? Some combinations of traits are good. They cost less, right? They're very favored. But whether you will actually grow or die is determined by this, but also by this. And this is determined, this is the ecological dynamics of competition for the resources. So as somebody becomes successful and they deplete the resources, that reshapes what landscape the others see. And that's what people call snowscape. And that is one of the interesting things on this model. I think it's one of the sort of minimal contexts for something like a snowscape with this kind of like flavor of real biology, which could, if it's something about this is solvable, that would be very interesting.
So you mentioned a bit, but do you mind elaborating? What do you miss biologically by the JJ being symmetric? Are there phenomena that are known that, or is this like?
I would say the limitation here is not that they're symmetric.
Automatically.
It's automatically symmetric because this, right? I can always.
But isn't it because you assume that you have the Lyapunov function that symmetry?
No, this is separate.
The Lyapunov function is assuming the two sigmas in the two equations.
Yeah. This is what creates the Lyapunov function. That's I use the same sigma.
They're perfectly efficient.
Yeah. Here, this is absolutely no problem, assuming that's symmetric, right? That what I'm missing here, the limitation is that this doesn't depend on mu. And in real life, of course, just how costly it is for me to run nitrogen fixation in presence of oxygen depends a little bit on I've evolved over time and I tried to make that better and I found some solutions, right? So that may depend on the strain. Yes.
Related to that point, are there like measurements or experiments that have shown that, for example, the same bacterium that switches phenotypes grows more slowly in one condition or in the other?
Yes, there are examples of that for sure. Generally, if you are in an environment with you can eat glucose, but you're in an environment where there's no glucose, you'll turn off the pathway making enzymes and receptors and transporters to bring in glucose because that's just empty cost because there is no glucose. And once glucose arrives, you'll sense that and you'll turn on those costly pathways to bring in the food, right? Just as a simple example.
Whether there's oxygen or not, that's
yeah.
general one.
Yeah. Okay, so
Question: there's a funny thing about once you add the evolution, right? If you don't have frustration, then you just get one thing that wins everything, right? You align yourself. If I didn't have the second order term.
Yes, but that's why you have the second order term.
Yeah, but it's weird. So it's really we played a lot with this, but it very depends on how frustrated that JI is.
Yeah, there's a lot to talk about there, which I'm not prepared to do right now, but this is definitely one of the things that. So in some sense, like I'm trying to be mindful of the time and I'll run out very soon. I wanted to show you some sort of cool phenomenology in this model, but in some sense, I'm happier to, as long as I explain this model to you and in what way progress here would approach some question like this, I think my duty is done. And then anybody who feels like you can contribute here, like I can tell you so much more about what we see here, right? Maybe I'll have time to give you a glimpse of what's going on, but yeah. So let me tell you how this fits into the broader sort of context of models, right? We didn't invent consumer resource models. We didn't invent bit flips. So what's the sort of interesting, how does this connect? There, in a lot of models, the fundamental object that you think about, why does it look the way it does, right? So models like resource competition models live here. They say, let me instead say what species do. So my fundamental object is some postulated object often called sigma mu I. We've seen this in some of the previous talks. Where for every species, every phenotype mu, I tell you which traits it has. For example, in the case of consumer resource models, which resources this organism is competing for. And then that determines how the species interact, right? So now this is a derived object because the species that is competing for the same resources, I'm a strong competitor with them, right? And somebody who does something completely unrelated to me, I don't see them. Right? So I postulate this object, and I want this to be then derived. What I'm presenting you here is doing one more step where I say, well, I'm not happy with postulating sigma mu I as my fundamental object either, because I want to know why do the phenotypes look the way they are, right? Which traits they actually carry, what's the reason for that? And so the fundamental object I postulate here is this Jij, which describes how traits interact, which traits go together well, which traits go together poorly. And then I let...
It's really the chi-I and the Jij. I mean, you have a cost also for each.
Oh, yeah. I always set all these to ones and, mostly think about this, but of course you're absolutely. Yeah. I should be including that in there. And so this is what I call evolution, or turning on pathways, turning off pathways, because this sets up some landscape of combinations of traits that are good, right, for objective reasons or for reasons of what's available, right? And then that determines kind of as my organisms navigate the landscape, turning on and off various traits. And once somebody establishes, they reshape this landscape for everybody else by depleting some resource, right? All that stuff, the eco-evo feedbacks, that will determine who lives in my community, and that will determine how they interact. Yeah.
So here okay, now but landscape. Landscape for me is a function.
Yeah.
Function of what?
Function of the phenotype. So for me, this growth rate of phenotype mu in a given condition. So what it's is a function of the environment, right? Environment is the ensemble of our eyes, which the community itself shapes, right? And mu is encoded in this vector sigma mu, which is sigma mu I, which traits this organism has. That's my kind of landscape, and it has this portion, which is fixed, shaped by these JJs, and that's why I'm saying it's actually a snowscape, because this thing.
Isn't there a Lyapunov function under the evolution sigmas as well?
Yes.
So there is
in this version. Yes.
You could write down.
Correct. And that is a thing that in a more general would disappear. But what are the first questions I want to ask here? And to connect this back to this implications of constraints. So let me go back to my example. Yeah.
Question. But it's also a technical question. To what extent do you think it matters that the constraints are soft and not inequality constraints? This is something that always bothers. Right here, like you're just good enough.
Soft, but not
I mean, they're not inequality constraints, right? They're like, you can imagine if I'm doing like,
right,
inequality.
I just have to be good enough at this.
You're right.
Oh.
You just have to be better than the other guys.
Yeah.
I'm good at that.
Yes, that's an interesting, questions I'm asking already exist here. That's a very interesting question. I think would be fun.
I'm wondering if you thought about it.
Yeah, no, I haven't thought about it. So let me give you an example of like connecting this back to the kind of question I want, which is how are community behavior shaped by physiological constraint? Let me go back to this example of nitrogenase is killed by oxygen, right? So this is an example that lives here. Example, nitrogenase is killed by oxygen, which means that in this matrix, if there's a trait, I have a large negative entry for the two traits can fix nitrogen and respires oxygen, right? Okay, so my question is, imagine that you're in a world where that is the only strong constraint and everything else kind of like is anything goes, right? I want to know, what does that, do I get any predictions or expectations about community behavior? Now let me give you an example intuitively that you might expect that to be true. So let's say I have a vat. I have a vat, and there are some microbes. Sorry, you guys can't see this. This is a very important diagram. So I have some microbes, some community living in a vat. I have no idea who they are. But now I sort of bubble oxygen, and one property that they're doing, they're fixing nitrogen at some rate. Now I bubble oxygen through my community. What do I expect? Well, I expect aerobes, the bugs who like oxygen, to increase. Maybe. And maybe one other thing I expect is the nitrogen fixation to go down because whoever is happy with oxygen, they're the ones who are not happy with nitrogen. And therefore, right, this could be an expectation. This is an expectation about the function of this entire vat without knowing anything what was inside. Is this true? Is this true? And this is in a simple world where there's no other interaction. And of course, that's not our world. So the second question is, what if many Jij interact? And, some sparse, strong constraints, and then they start chaining and interacting and frustration and things like that, right? Okay. Now, I am out of time, even with the 5 minutes of the elevator traffic jam. So let me just announce to you a couple statements, and we can talk more in a sort of discussion format right before lunch. There'll be some room for... I don't think this is the right context for the discussion, so we'll just do it in a corner of the open space. It kind of feels more comfortable to talk. So let me just announce to you 2 results. If I actually do this world with just one strong constraint, and I think about this experiment, I add resource 1, trait 1 and trait 2 interact very negatively, what happens to the total flux of resource 2? So aerobes go up, that's true. The flux of resource 2 is absolutely perfectly buffered. It does not go down at all, because in a world where this is the only interaction, everything else is very flexible, and this has been showed before. Lorenzo Font et al. And with Jacopo, and I work with Remy and many others. Pankaj. So that would kind of contradict that intuition. So where does this intuition come from? Well, there is a regime where this intuition is true. If I have many strong interactions, that actually creates the kind of compositional rigidity to the community, and that is what you need so that if you promote everybody who has trait 1, they are depressing through competition with all other resources the guys who are eating trait 2. So in the regime of many interactions, this is indeed the intuition you get. So this kind of what seemed reasonable secretly is actually the regime of many interactions. And you can define here two reasonably different, qualitatively different regimes of response to perturbation, which I call shallow wells regime and deep wells regime. In shallow wells, you push on the system, and interactions are mostly weak, and everybody can adjust what they're doing to buffer the perturbation. In fact, buffer it better and better as you make the perturbation stronger within some limit. Whereas this deep well regime, where there's many strong interactions, you can just change what you're doing. There are rabbits, there are foxes, and foxes don't, mutate into rabbits. Which is why if you depress the rabbits, you affect the foxes in some predictable way. And so these regimes have different properties dynamically, and that's the kind of stuff that we can do simulations about, but also it seems very approachable analytically. And I think there's lots of good questions, which I'll try to pitch at you in the more topical discussion for those who are interested. Thank you, and sorry for being over time. Yes.
We know about the number of strong interactions that you have.
Excellent question. So these JJs that I said, they're JJs. So my general pitch to you is that JJs exist in real life, and they may have consequences for community behavior. What I described to you here is a program where you postulate some JJs, and you see what you can understand about here, right? If you understand this well enough, you can also presumably maybe ask the question of, given some real-life observations, can I back out what these JJs are? And one, I think, very interesting thing is actually to look at the structure of these matrices and try to back. One of the very concrete and very interesting questions is, how does structure of JJ shape the structure of sigma? Because this could be potentially accessible with something like the metagenomes of bugs, which traits they carry. Can you infer from there and, in fact, disentangle the kind of constraints versus the environment? Are you carrying these traits because they don't go, yeah.
We do not have control over how should be the JIJ matrix.
We don't have control here. So for me, JJ implements the universe. And sadly, I don't actually have control of the universe. But
evolution. A lot of it's the constraints coming from evolution. The fact that nitrogenase doesn't like oxygen is probably because of oxygen.
Yes. So here I'm separating kind of.
Nitrogenase.
I'm assuming there's some sort of separation of time scale in evolution, which is there are some things that are kind of like hard constraints to get around, and that's the JIJs, and they don't change, at least on my scale observations. And then there's my evolution here operates at the level of the sigmas, which is the bugs changing what they do, trying to contend with that. And of course, ideally, you should be somewhere in the middle. That's the next step. That would be very interesting. Yeah.
What is the dynamics of the sigmas?
Yes. So dynamics of the sigmas are, so in a simulation, I can tell you what I do, which is that I start with a random phenotype, right? And then I do a proper continuous time and discrete event Gillespie sort of loop where there's the continuous dynamics of ecology, and every mutant has some, invasion rate, which you can properly population genetics treatment, turn into the probability of it actually escaping drift and getting introduced into the population, and those are the dynamics. I don't think you can do analytics in that regime. That's where the existence of a Lyapunov function can really help you, because then the sort of equo-evo equilibrium to which you arrive, which is actually what, if I assume that I can just run it to forward time to equilibrium, this optimizes some Lyapunov function, which is how I should be able to access properties of it. That's why for analytics, that would be a benefit. And maybe some smart people know how to do it even without that. I know they can. I can't. But how I know we're so way over time, so the chair will stop me.
And so are you saying that this dynamical system, if there's no evolution, will typically converge to a fixed point?
Even with evolution.
Well, yeah, but as long, for a fixed set of sigmas, right? So if you run the dynamical system,
yes,
it will be some stable composition, and then you can sort of perturb it by evolution.
You can do it that way as well.
So there's no, chaos, not none of that stuff that Tina was talking about.
Absolutely. That's a different, yes, that's that. And for my questions, I find that helpful because, even here these questions exist, and are reasonably non-trivial and interesting. And of course, if everything here, these dynamics were chaotic, but that's why we have Valentina, who studies that regime, and I study this regime, which is much nicer, but I can ask these other questions. I see Karthik in the back.
Yeah, I was wondering if this buffering and like the shallow well and deep well properties are happening with still space for evolution, or like have you fixed the sigma mu's, and then you're trying this increase in oxygen experiment and seeing that they're still buffered?
Yes, that would also be fun. We should talk about this. So again, there'll be another time for additional discussion just before lunch. I think we should stop here because so let's have our break and coffee, and I'm available for any questions.